A patient arrives at the hospital with profound fatigue and tingling nerve damage in their extremities. Blood work reveals a catastrophic failure. Their white blood cell count has completely crashed, leaving their immune system entirely offline. Oncology performs a bone marrow biopsy to examine the body's cellular factory. Under the microscope, they find highly abnormal, vacuolated cells, shattered scaffolding that leads to a diagnosis of myelodysplastic syndrome, a severe, life-threatening blood cancer. While the marrow looks cancerous, a secondary test for serum copper reveals a near-total deficiency, a finding that contradicts a primary bone marrow failure. The investigation shifts from the pathology lab to the patient's daily routine, uncovering the culprit inside their medicine cabinet. Over-the-counter zinc cold lozenges, taken daily for months, have triggered a cascading system failure. Flooding the digestive tract with massive doses of zinc forces intestinal cells to manufacture a buffer protein called metallothionein. But this protein has a high binding affinity for copper. It acts like a biological magnet, indiscriminately grabbing and locking down dietary copper, which blocks it from entering the bloodstream. Without copper, bone marrow cannot produce mature white blood cells. Trace minerals operate on a razor-thin homeostatic tightrope, disrupting the balance. To understand why the body relies so heavily on this specific element, we have to look back to the primordial ocean. Following the Great Oxidation Event, newly dissolved zinc became available to early eukaryotic life, providing a stable alternative to the reactive metals of the deep. This selection was a matter of subatomic physics. Unlike highly reactive metals like iron, zinc possesses a completely filled outer electron shell, known as a D10 configuration. Because its outer shell is entirely full, the zinc atom operates like a perfectly balanced, rapidly spinning top. It does not attempt to steal electrons from other molecules, nor does it easily give them away, preventing it from undergoing the dangerous, cell-damaging Fenton chemistry associated with iron. This lack of volatility makes zinc a reliable structural material. It provides the stability needed to act as the master anchor for the human genome, holding complex systems together without the risk of an accidental oxidative explosion. This stability makes zinc the foundational, non-explosive structural architecture required to build and sustain complex life. A healthy adult contains roughly 3 grams of total zinc. However, the body doesn't maintain a free-floating reservoir. 90% of that reserve is locked away tightly, sequestered deep within the mineralized matrix of bones and skeletal muscle. Less than 0.1% of your total zinc circulates in the blood at any given moment. To keep the system running, we rely on a constant influx from high-density dietary sources, with oysters providing 50 to 100 milligrams per serving. Once inside the cell, this metal acts as a structural anchor. In a motif known as the zinc finger, a single zinc ion forces a short protein chain to fold into a rigid loop. These microscopic fingers slot into the major grooves of our DNA, allowing the protein to read and regulate gene expression. The body also uses this rigidity to guard volatile hormones. In the pancreas, 6 individual insulin molecules wrap themselves around 2 central zinc ions to form a stable storage vault. This prevents the hormone from degrading prematurely, ensuring it remains functional until the moment it is needed to manage blood sugar. From organizing the genetic code to stabilizing insulin for storage, zinc provides the rigid structural anchor these high-stakes systems require to function. When the gates regulating this metal fail, the consequences are immediate. In a genetic mutation known as acrodermatitis enteropathica, an infant possesses broken ZIP4 transport channels. Once they are weaned off maternal breast milk, they become physically incapable of absorbing dietary zinc. Without the zinc scaffolding, tissue repair enzymes fail entirely. The infant develops severe skin erosions around the mouth and eyes, and the lining of the gut physically dismantles, leading to immune failure. While genetic flaws starve the gut, an occupational hazard bypasses the digestive system entirely. Metal fume fever is an acute inhalation toxicity seen in welders working with galvanized steel. The extreme heat of a welding torch vaporizes the zinc coating into ultra-fine oxide fumes. These deposit deep inside the lungs, triggering a panicked cytokine storm. Because the metal is embedded in the tissue, there is no molecular antidote. The body must simply clear the metallic debris over 48 hours. Homeostasis demands exactness. A lack of dietary zinc leads to physical disintegration, while inhaling the vapor triggers an explosive immunological panic. Measuring the tiny fraction of circulating zinc requires intense precision. We have moved from historical manual lab benches to inductively coupled plasma mass spectrometry, a process that atomizes blood inside an 8,000 Kelvin plasma torch to count individual zinc atoms. But the measurement is easily compromised. Zinc is an abundant industrial element, present in airborne dust and the rubber stoppers used in routine blood collection tubes. If blood is drawn into a standard tube, the rubber stopper immediately begins leaching zinc into the sample. To prevent this, phlebotomists must use a specialized, acid-washed royal blue top trace element tube. Physical stress can also ruin the results. If the phlebotomist uses a needle that is too small, the shear stress will rupture the red blood cells as they enter the tube, a process known as hemolysis. A single red blood cell contains 15 times more zinc than the surrounding plasma. This physical rupture creates an artifactual toxic spike on the lab report, even if the patient's actual levels are normal. The most high-tech analytical machine in the world is clinically useless if the act of drawing the blood introduces invisible chemical contamination. Sometimes the body intentionally deceives the doctor. During a severe infection or massive trauma, the immune system releases a distress cytokine called interleukin-6. This signal commands the liver to act as a massive vacuum, instantly pulling circulating zinc out of the bloodstream and hiding it inside hepatic tissue. This is a tactical defense. By pulling the zinc out of the blood, the body is intentionally starving invading bacteria of their resources they need to replicate and survive. This creates an inflammatory illusion. If a physician misinterprets this low lab result as a nutritional deficiency, aggressively administering zinc could inadvertently feed the bacterial infection the body is attempting to halt. This brings us back to the misdiagnosed cancer from the emergency room. The cure was not chemotherapy, but a profound respect for this balance. Once the patient stopped the zinc lozenges and began copper replacement, the intestinal trap released, and their blood counts normalized in weeks. Understanding the Master Sculptor requires observing the microscopic balancing act that connects the environment around us directly to the DNA inside our cells.